Hello, my name is John Teodosio, and this is my research on the impact of Leptoglossus phallopus population density on free-living symbiont Caballeronia. With the growing global population, agriculture has become more important than ever. There has been increased interest in understanding agricultural pests and how to control them to protect crop yield. Several important pest species are true bugs, such as Leptoglossus phallopus, and engage in promiscuous symbiotic association with the free-living soil bacteria Caballeronia. Caballeronia is a free-living soil bacteria, so it does not depend on the insect, but the insect depends on Caballeronia for its survival. Phallopus acquires the bacteria from its environment every generation in its early developmental stage called the second instar. Caballeronia is necessary for the bug because it colonizes the gut and grows to high abundance. The insect digests excess cells, obtaining B vitamins and amino acids. Recent work has shown that live Caballeronia cells are transmitted to the environment through the feces of Anassotristis, another pest in the same family. Our research sought to further describe the bug Caballeronia symbiotic association and agricultural impacts thereof. We sought to answer the question, how does bug density affect symbiont transmission to soil? We hypothesized that the soil microcosms exposed to higher densities of bugs would be colonized with more Caballeronia, the free-living symbiont. To test our hypothesis, we inoculated second instar nymphs for 72 hours with LEP1A1 strain of Caballeronia. LEP1A1 was used because it commonly associates with Phallopus in natural populations. The nymphs were transferred to sterile soil microcosms with southern cowpea plants and peanuts to subsist. There were 4 treatments, one with 0 bugs, 5 bugs, 15 bugs, and 30 bugs. The bugs were reared to adulthood and soil samples were taken from each treatment. Then we did soil fecal DNA extractions on the soil samples to extract Caballeronia DNA. The DNA was quantified with qPCR using specific primers that target the LEP1A1 strain of Caballeronia. Additionally, we treated one set of final soil samples with PMA to remove dead cells in order to determine the amount of live cells in the soil. We ran statistical models and found that the Caballeronia abundance increases with increasing bug population density. In the results, the y-axis is the mean Caballeronia abundance detected by the qPCR. The final Caballeronia abundance, shaded in green, was the highest, with the highest bug population. Our data suggest that the bugs are releasing Caballeronia into the soil. To determine whether these were live cells being quantified, we treated soil samples with PMA, which binds with dead cells' DNA and removes them, so downstream analysis, like qPCR, only quantifies live cells. We did not see a significant difference in Caballeronia abundance at different bug densities. So, we cannot conclusively determine that the higher abundance of Caballeronia at higher density bug exposure was due to live cells being excreted. This may be because PMA was not specifically made for use in soil samples and may not have been as accurate. However, we are currently running a follow-up experiment where we place second instar nymphs in the same soil microcosms after removing the adults. We are measuring their development time to see if they can acquire live Caballeronia cells from the environment. Development time is used to measure this because bugs lacking the symbiont develop much slower and often do not develop into adults. If Phallopus migrates to a new area and transmits the symbiont to the soil, this could cause a pest population boom because the next generation of insects could more easily acquire the symbiont. Understanding the bug Caballeronia symbiosis can inform the development of more effective pest control strategies that account for Caballeronia's role in the proliferation of Phallopus. More work is needed to expand on our understanding of this symbiotic association. This could include doing DNA extractions on leaf tissues of plants in the microcosm to evaluate how the symbiont is transmitted to other portions of the bug's environment. I would like to thank the U.S. Department of Agriculture and the University of Texas at Arlington for their support of this research. I would also like to thank my mentors, Vivek Singh Parajuli and Dr. Allison Ravenscraft.